Hi, I'm Zachary Baker.
And I'm Jessica Myers.
And welcome to Geneticist News. The story of the hour is all about DNA.
The molecule's full name is deoxyribonucleic acid. This molecule is a nucleic acid that contains the genetic instructions for the development and function of living things.
All known cellular life and some viruses contain DNA. The main function of DNA in the cell is long-term storage of information and genetic coding.
DNA will actually expand out to be about 6 feet. But DNA inside our bodies is condensed down so far it can fit inside of a microscopic cell, never to be seen by the naked eye.
So how was it that we came to know about this tiny molecule? Who discovered it? How is this molecule arranged?
All this and more next on Geneticist News. And we're back with the story on how it all began.
And now to our history correspondent, Jacob Stinchcomb, with the story.
Hi, I'm Jacob Stinchcomb, and today we are in the history section of our DNA Geneticist News. Today we will refer to many historical backgrounds on DNA, starting with Gregor Mendel, the father of genetics, as we like to call him, a very famous man, performed several experiments with plants in 1857 that led to an increase in the study of genetics. He was the first to notice how inheritance worked as he experiments with several pea plants and noticing different changes with the descendants of his first pea plants. However, becoming a monk in the Roman Catholic Church, he was then told to give up his research, and from there, Mendel no longer helped science. But that was not the end for DNA's history, as it went on to Friedrich Miescher and Richard Altman. Friedrich Miescher discovered a substance that he called nuclein in 1869. He discovered this in a gruesome way. He found dead body bandages from hospitals, and discovered from white blood cells in those bandages a gray silky substance. And not knowing what on earth this was, he now began to call this new substance nuclein, which then became nucleic acid. Richard Altman was his pupil at the time, and he is the one who did rename this as nucleic acid, which is much more scientific for us greater minds. Last but not least in this timeline, we have Rosalind Franklin. Rosalind was a very brilliant woman and tried to make this crystal of the DNA molecule. She was responsible for how we believe the structure of DNA is. A long time ago, when all of you were not born, and this was back in the 60s and 70s, back in the old days, DNA was believed to be a spiral structure, almost like a Christmas tree with lights, so to speak. However, thanks to several scientists down the line, such as James Watson and Francis Crick, we then began to understand the true nature and structure of DNA. Through history, we now know that it is actually a double helix and not a Christmas tree with lights. Yes, sir, because of these great historical discoveries, we now understand DNA to be something very important. And now, that concludes our history examination.
Hi, I'm Jessica Myers, and we're here with interviews with the general public to see what the public actually knows about DNA. What do you think is the function of DNA? This sucks. Hi, I'm Jessica Myers, and we're here with more interviews with the general public. Let's see what this civilian, Helen Keller, has to say about DNA. What do you think about DNA?
I think that DNA is a myth, you know? It's like in the ground and the grass in the air, but it's not really real.
So it's not real, but it's in the air.
Yeah.
Fascinating insight. Thank you, Helen Keller. Hi, I'm Jessica Myers, and we're here with more interviews with the general public to see if anyone actually knows what DNA does. All right. Hi, civilian. What do you think DNA is, and how does it function?
Well, I think that it's the basic structure of how our
So how our body functions through genetic coding, and it is a double helix structure with nucleotides with cytosine, thymine, guanine, and adenine, and it connects through phosphate groups.
Oh, wow. I think we actually know someone who has some stuff to say about
Nobody cares.
All right. We'll be back with more interviews with the general public later.
Thank you, Jessica. And actually, I have a pretty big surprise for you today because DNA Polymerase here actually agreed to accommodate to allowing me to join him in one of his typical training runs. So I'm pretty excited. I hope you guys enjoy this.
You ready?
Oh yeah, of course. But well, this is something else I can.
You good? You
Yeah,
okay?
yeah. Good, thanks, Polymerase. Okay, so before we get back to Jessica in the studio, I have a couple questions for you, Mr. Polymerase, if you don't mind.
I don't mind.
So, one of the main questions that a lot of our viewers have been dealing with, one of the main questions that's been boggling their heads for a long time is how are you actually handling your new position on the team in a lot of the repetitive replication?
Well, we all have our parts on the team, and each one is just as important as the other. My position in the helix is structural, but what I really hope to accomplish in the upcoming replication is just to keep the stress off the proteins, because the less misstructures to fix, the better.
Thank you, Mr. Polymerase. And regarding that statement, is that in any way related to the recent criticism that your teammates in the Kelvin cycle of the team have been giving you recently?
Yeah, they're expecting a lot less misreplications by the end of the G2 phase. So if I can just push through G2 till the break in the third quarter, we might be able to beat our team's record replications time, with there being fewer time penalties for the misreplications.
I understood that does sound like a lot of stress that's being put on you recently. Yeah. Okay. Well, there you have it, Jessica. Back to you in the studio.
Fascinating story, Isaiah. I'm sure Zach will be pleased to hear it as soon as he gets back from, well, never mind. But we do have a story for you on the proper function of DNA. DNA is essential to life. Without it, we would all cease to exist. DNA's essential function is to store genetic information that tells organs how to function. In humans, half of the DNA for a child comes from the father and half from the mother. It is able to replicate itself from new cells because of the double helix structure. Each nitrogen base on the DNA strand is only able to pair with one other nitrogen base. Therefore, when it splits, the DNA knows what to pair with for each nitrogen base to complete a new strand. That is how DNA works.
Oh, sorry, I didn't know it would take so long.
Oh, hi. You just missed the story of a lifetime, Zach. But, oh well.
We have an interview with a highly specialized biologist, Jacqueline Keating.
Let's hope she gives us some special insight into her version of the central dogma of biology.
Thanks, Zach and Jessica. I'm here with biologist Dr. Jacqueline Keating. Thank you, Doctor. It's a pleasure to be here. So, what exactly are you studying? Well, I have discovered the central dogma of biology. So we know that the cell is the basis of all living life. But why is it the basis of all living life? So we have DNA, but how does it replicate and transcribe and translate into RNA and protein? So how do DNA, RNA, and DNA just connect? Well, it's the replication of DNA. What is replication? Well, replication is the double helix is unwound and each strand acts as a template for the next strand. Bases are multiplied to synthesize the new partner strands. It is the biological process of producing two identical replicas of DNA. So by the end of this, we'll have two replicated copies of the double helixes. and then it transcribes. This is the first step in gene expression in which a particular segment of DNA is copied into RNA by the enzyme polymerase. Both DNA and RNA are nucleic acids when they use bases pairs of nucleotides. So by the end of transcription, we will have single strands of RNA. And then it translates into cellular ribosomes to create proteins, messenger RNA produced by transcription form DNA. It is decoded by a ribosome to produce a specific amino acid in chain or polypeptide, which is protein. And this is the central dogma of biology.
And there you have it, the central dogma of biology.
Thank you, Catherine. And what an interesting theory, Jacqueline. Thank you for your dedicated research.
Our journey through the complex molecule of DNA is almost finished.
Oh, but we do have one more story for you. In the Netherlands, a man is designing and building a new hotel to model after the structure of DNA.
And now we go to Catherine with the story.
Thank you, multimillionaire Caleb Daniels, for joining us today. I understand that your company, Helix Incorporated, has a new method of building structures of DNA.
Please, Catherine, it is not multi-millionaire Caleb Daniels. It's billionaire Caleb Daniels. Multi-millionaires were so last year.
Well, what exactly does Helix Incorporated have for us today?
Oh, you're in for a treat. My company has created a new innovative way of DNA-classed hotels that include the double helix structure, named after my company.
So, how do you build these new DNA hotels using the new helix structure?
Well, Catherine, it's hard to describe in a detailed way, but I will say that it involves several groups, including the phosphate group, the sugar group that everyone knows and loves, and not to mention nucleotides that help keep this structure going.
So, rumor has it that you'll have nitrogenous bases involved.
Oh, you've heard it right, Catherine. There are 4 nitrogen bases. Not 1, not 2, not 3, but 4. 4 nitrogen bases for the public to enjoy. We got adenine, guanine, thymine, and cytosine. All 4 of these will help making this structure highly compacted together with the coating that is required for such a hotel.
It looks very impressive.
It is very efficient, my dear.
So, rumor has it that you are building on a smaller scale, more for the people as well.
Yes. The DNA Strand Hotel is a monument to Helix Corporation's work. But we need something that the entire people can see. That is our motto, after all. Helix reaches all, including the people. So, in light of that, we have created a new single-strand hotel known as RNA. It is not the same double helix strand that we have here, as this model shows, but we do have a single strand, which is more suitable for the people.
So, I hear that you'll still have the famous nitrogenous bases involved.
Indeed we do. We still have 4 nitrogen bases, but 3 of them are the ones that we'll see here. Adenine, guanine, and cytosine. But however, instead of thymine, we'll be using uracil instead for the RNA structures.
Thank you, Mr. Daniels.
And that's all we have for this hour of Geneticist News.
I'm Zachary Baker.
And I'm Jessica Myers.
That's all, folks.
On Geneticist News.